scholars and students. Good morning to everyone. On the behalf of the Organization Committee for the 3-day National Symposium on Trends and Innovation in Pharmaceutical, Biochemical and Biosciences, I, Pratyancha Sharma, Organizing Secretary of the Symposium, feel honored to extend a warm greeting to you all. Now, I would like to invite Dr. Amit Sharma, the convener of the symposium, for welcome address. Over to you, sir.
Thank you, ma'am. Thank you so much. Hello. Good morning to all of you. Hello and welcome once again. Today is the third and the final day of the National Symposium. I welcome all once again, all the dignitaries, eminent speakers, and all the participants from different parts of the nation and outside India. I feel very proud to announce here that the National Symposium received a very great response from all over India, and people are appreciating the academic platform sponsored, recognized by the Formalo. I remember one special quote from my one of the greatest mentor. हम चले थे बड़ी शिद्दत से अकेले ही इस बड़ी दुनिया में. हम चले थे बड़ी शिद्दत से अकेले ही इस बड़ी दुनिया में. लोग मिलते गए और कारवां बनता गया. लोग मिलते गए और कारवां बनता गया. I welcome Honorable Chancellor Engineer S.K. Punj sir, Chief Managing Director Ms. Tripta Punj ma'am, Pro Chancellor Engineer Tushar Punj sir, Honorable Vice Chancellor Professor Dr. R.S. Rana sir, Pro Vice Chancellor Professor Dr. N.N. Sharma sir, Registrar Professor Dr. Jahid Ali sir, Dean Engineering Professor Dr. Raminder Pritpal Singh, and he is also our technical and moral advisor. Today, we have the three eminent speakers, Professor Dr. Mahinder Singh Ashawat sir, Laureate Institute of Pharmacy, Himachal Pradesh, Professor Dr. Grish Kumar Gupta sir from Sri Sai College of Pharmacy, Badhani, Pathankot, Dr. Muhammad Mazhar from K.R. Mangalam University, Haryana. They have very vast experience in the field of pharmaceutical and pharmaceutical chemistry. I believe that the session will be very informative for all the participants, and I also believe that these 3 days would be acknowledgeable for all of you to applicable in different way in your life. I thank one and all my teammates who sincerely work to make this national symposium a great success. The credit goes to all the teammates. I also thankful to the management and the higher authorities of the university to continue sports and motivation. I especially to my colleagues to act as a moral booster to work rigorously towards success of the event. Thank you to all once again. Thank you very much. Jai Hind, Jai Bharat.
Thank you, sir. Now, behalf of the team, it gives me a great pleasure to welcome the Professor Dr. Mahinder Singh Ashawat. He is presently working as Director cum Principal at Laureate Institute of Pharmacy, Himachal Pradesh Technical University Research Center. He had been associated as an honoring member Board of Studies, Academic Council member, Research Board and several committee member at Himachal Pradesh Technical University. As an academician, he has recognized as the Best Teacher Award and twice of Best Research Paper and Speaker Award in national conferences. Sir has been awarded as Principal of the Year Award 2017 by Society for Pharmaceutical Education and Research. He had been member of international societies for various international conferences. The major focus of his research interest is divided into different segments of research parts, intradermal depots, herbal cosmetic, and their mode of action. Dr. Ashawat is also serving as an editor board member of various journals and sprints. He has published 8 books and one edit chapter, more than 100 research and review paper in international and national journal, and presented more than 50 paper in conferences and seminar at international universities. Dr. Ashawat had been served as state vice president of APTI, moreover assuming overall responsibility of a devout educator, covering professional development, member of various committee of staff, state technical university, concerned to affiliate and staff and election committee. May I now request the Professor Dr. Mahendra Singh Ashawat sir to address our esteemed participant on the National Symposium with his topic Modern Biological Tool Cosmetic Dermal Formulation. Over to you, sir.
Thank you very much, Prichansi. Hello, everyone. Namaskar. I would like to first thanks to God Almighty Jwala Devi. I would like to thanks the organizer of this National Symposium on Trends and Innovation in Pharmaceutical, Biomedical and Biosciences at this university, Science University, Palampur, for inviting me here today. So very good morning, everyone. Welcome to this presentation. And first of all, let me thank you all coming to this symposium. There are certainly many reasons. That's why I am among with you. The first reason, because of the patron sir, S.K. Punj sir, chancellor of this university, and the next pious reason, the presence of Professor N.N. Sarma sir, pro vice chancellor of this university, and next to this, my fellow teacher, Dr. Amit, Dean Pharmacy, and utmost reason to become with you is our beloved and most enthusiastic participants in this symposium. So I thank you all for inviting me to interact with you all, because it's an opportunity for me. So in this today's presentation, I just designed a one specific presentation for you, because that is uh, meant for your purpose only. Because you know there is so many medicines are innovating day by day, but there is a team of scientists are working on specifically dermal formulations. And as I think everyone among we people are using the dermal formulation either in form of the cosmetics. So today I prepare this particular presentation with you. I am just sharing my screen. Is this visible to all of you?
No, sir. Sir, now it is visible.
Now it's visible.
Yes,
So
sir.
are you able to see this title of this particular presentation, Modern Biological Tool Cosmetic Dermal Formulation? Is this visible or not?
Yes, sir.
Right. So as title specifically reflect the its content, that dermal formulation, and what kind of novel tool can be used in the research of dermal cosmetic formulation. Today I just want to share whatever knowledge I possess in my research time period or research tenure. I just want to share with you. So as we all know, as we all are using since beginning the cosmetic formulations. कॉस्मेटिक के बारे में आप सभी लोग अच्छे से जानते हैं, और सबसे बेस्ट यूजर यूथ ही है आज की डेट में, क्योंकि कॉस्मेटिक की जरूरत हमें हमें सबको ही पड़ती है. सो फर्स्ट लेट मी क्लियर द व्हाट इज द कॉस्मेटिक. जनरली हम कॉस्मेटिक को सिर्फ क्रीम और लोशन और पाउडर से ही डिनोट करते हैं, बट राइट नाउ द कॉस्मेटिक मीन्स आर बिकमिंग चेंजेस, राइट. So I just want to put that its definition, general definition, जैसा कि आप लोग जानते हैं, any substance used to clean, improve and change the complexation of the skin and improve the luster of hair, nails and teeth. जैसा कि हम अभी तक जानते हैं, बट द कॉस्मेटिक फॉर्मुलेशन इज वेल क्लियर डिफाइंड इन आवर फार्मेसी एक्ट. So as far as concerned with the Drug and Cosmetic Act 1940, it is defined that cosmetic is considered as an article intended to be rubbed, to be poured, to be sprinkled or spread on another, introduced into or otherwise applied to the human body or any part, therefore cleansing, beautifying, promoting the attractiveness or alerting the appearance, and including any article intended to the use as a component of the cosmetic. इसको हम कॉस्मेटिक बोलते हैं. अब आप देखिए कि कॉस्मेटिक आप सभी लोग यूज करते हुए आए हैं. हमें क्यों कॉस्मेटिक की जरूरत पड़ती है? ये कंप्लीट दिख रहा है आपको?
Yes,
Now,
sir.
yes. So why we are so much we can say need, why we are using such kind of cosmetic? So as we all know, our skin is organ which is directly exposed to the environment, and it comes into the so many different kind of I can say insulting environment like UV radiation, microbes, dust. So many external factors affect the health of the skin. And skin, you will be surprised, this particular organ, skin considered as an organ. So this particular organ is very miraculous. It's having its system. इसका अपना खुद का मेटाबॉलिक सिस्टम होता है, इसका खुद का रेस्पिरेटरी सिस्टम होता है, इसका अपना स्किन का अपना एक्सक्रीटरी सिस्टम होता है, राइट? सो it protect from heat, cold and any kind of pain perception which are initially felt by skin through the skin surface. Is it? आप देखिए कभी भी स्किन के ऊपर अगर हल्का सा भी इचिंग होती है तो वो इचिंग क्यों होती है? क्योंकि आपको कहीं ना कहीं स्किन ने इन्फेक्टेड किया है, कोई ना कोई माइक्रोब्स आपकी स्किन को अफेक्ट कर रहा है. सो स्किन फंक्शन अगर आप देखें, इट प्रोवाइड द प्रोटेक्टिव लेयर और बैरियर अगेंस्ट द मैकेनिकल हैजर्ड्स, अगेंस्ट द थर्मल हैजर्ड्स, और वी कैन से फिजिकल इंजरी और हैजार्डस सब्सटेंसेस लाइक केमिकल इंजरी. आप देखिए हमारा स्किन जो है अपने आप में एक मेंटल सिस्टम है, इसका अपना एक पीएच होता है. लेकिन आपने देखा होगा, आपने एक्सपीरियंस किया होगा, स्किन जो है जब हम स्विमिंग के लिए जाते हैं, स्विमिंग करते हैं और कंटिन्यूअस स्विमिंग जब आप करते हैं, once your skin is exposed to the alkaline water, your skin became dry or wrinkled or whitening, isn't it? So the reason, because skin pH become alkaline. हमारा स्किन का pH जनरली क्या होता है? एसिडिक होता है. लेकिन वो जब आप एल्केलाइन सब्सटेंसेस और एल्केलाइन वाटर के उसमें रहते हैं कांटेक्ट में, तो आपका स्किन का जो पीएच है वो बढ़ जाता है. तो आप देखिए, बट लेटर ऑन आफ्टर few minutes, we can say within 10 or 15 minutes, your skin became again normal. तो ऐसा क्यों होता है? Because skin having its own pH mental, राइट? अपना पीएच मेंटल होता है, वो उसको बफरिंग सिस्टम उसको अपने आप वापस से एसिडिक पीएच पे लेके आता है. इट मींस स्किन इटसेल्फ हैविंग इट्स ओन सिस्टम, क्योंकि इसकी केमिकल प्रॉपर्टी जो बायोकेमिकल जो पैरामीटर्स होते हैं वो ऑटोमेटिकली उसको बेसलाइन पे लाके और स्किन को एसिडिक एनवायरनमेंट देते हैं. इज इट? इट प्रिवेंट द लॉस फ्रॉम द मॉइस्चर. आप स्किन का जो आपकी बॉडी के अंदर जो मॉइस्चर होता है वो भी स्किन के द्वारा कंट्रोल रहता है. डिहाइड्रेशन आपने देखा होगा, स्क्राउचिंग टेम्परेचर इन समर, इट अफेक्ट्स द डिहाइड्रेशन. इज इट? तो फिर हम क्या करते हैं? स्किन को प्रोटेक्ट करते हैं. इट आल्सो प्रोटेक्ट फ्रॉम द यूवी रेडिएशन. स्किन ही एक ऐसा ऑर्गन है जो आपकी आई पीपल्स और आपका स्किन, ये दोनों आपके इंटरनल वाइटल बॉडी पार्ट्स को प्रोटेक्ट करता है यूवी लाइट से. आप देखिए, आप यूज भी करते हैं सन प्रोटेक्शन फैक्टर बेस्ड लोशंस, जनरली हम समर में यूज करते हैं. द रीजन क्योंकि हमारी जो स्किन है वो यूवी से एक्सपोज्ड रहता है। हैजार्डस जो पोल्यूटेंट्स हैं जो हमारी एयर के अंदर हैं, एयरबोर्न पोल्यूटेंट्स होते हैं, आपके पेट्रोल और डीजल और स्मोक की वजह से जो पोल्यूटेंट्स जो होते हैं, सब्सटेंसेस, वो आपकी स्किन को क्या करते हैं, हैरेश करते हैं। और जब यूवी लाइट आती है, आप सब साइंस बैकग्राउंड से हैं, हमारा जो ओजोन लेयर है जो किलोमीटर्स का थिक है, लेकिन उसके अंदर ओजोन की वजह से जो लेयर है वो थिन हो चुका है, तो उसमें से यूवी लाइट जो फिल्टर होके आता है, वो यूवी लाइट जो होता है उसमें अलग अलग यूवी के क्लासेस होते हैं, यूवी ए, यूवी बी एंड यूवी सी। आपने देखा होगा कि जो लॉन्गर वेवलेंथ है वो 320 से लेके 400 नैनोमीटर तक का होता है। तो वो जो लॉन्गर वेवलेंथ है वो आपकी स्किन को बहुत ज्यादा अफेक्ट करता है, एंड देयर इज द वन ऑफ द मोस्ट कॉज ऑफ द स्किन कैंसर, क्योंकि वो लॉन्गर वेवलेंथ है, वो डीपर टिश्यूज पे जाके अटैक करता है, वहां म्यूटेशन करता है, एंड इट कॉजेज द skin cancer. So we can protect the skin from the external UV radiation by using different kind of cosmetic formulation. ये मैं आपको इस वजह से बता रहा हूँ क्योंकि जो हम आगे पढ़ेंगे, हमें कॉस्मेटिक फॉर्मूलेशन या डर्मर फॉर्मूलेशन कैसे यूज करने चाहिए, उसके बारे में, उनकी क्या क्वालिटी होती है, क्या हम वो क्वालिटी कॉस्मेटिक फॉर्मूला ही यूज कर रहे हैं या नहीं कर रहे हैं, इस चीज को आज आपके यहाँ हम शेयर करेंगे। It particular skin also regulate the temperature, right? And there is certainly reason ऐसा क्यों होता है, because skin is very complex. हमें लगता है स्किन बड़ा सिंपल है, but anatomically it seems a layer, but physiologically there is different changes, and anatomically once you will see inside the beneath of the skin there is different layers. So there is three basic layer, एपिडर्मिस, डर्मिस एंड हाइपोडर्मिस। एपिडर्मिस ही एक वो लेयर है जो कि एक बैरियर की तरह काम करती है, एक लेमिनेट की तरह काम करती है। इसके अंदर भी फर्दर 3 और 4 लेयर्स होती है। देन बट देयर इज नो ब्लड वेसल्स, देयर इज नो ब्लड वेसल्स। देन डर्मिस, देन हाइपोडर्मिस। आप देखिए हाइपोडर्मिस वो रीजन है जहां आपको सेंसेशन फील होते हैं, जहां नर्व एंडिंग्स होती है। डर्मिस वो रीजन है जहां आपके हेयर फॉलिकल्स होते हैं, आपके सीबम ग्लैंड्स होती है। ये जो है इसके डिफरेंट लेयर्स होते हैं जो इसकी फिजियोलॉजी को, इसकी बायोकेमिस्ट्री को रेगुलेट करते हैं। नाउ यू विल सी द एपिडर्मिस। सो एपिडर्मिस इज अपर मोस्ट लेयर ऑफ द स्किन इज स्ट्रेटम कॉर्नियम। स्ट्रेटम कॉर्नियम जो होता है, इट्स सिंपल डेड केराटिनाइज सेल्स होती है जो कि स्किन के मेटाबॉलिज्म से ही फॉर्मेशन होता है उनका, और वो सेल्स ऊपर आके एक केराटिन का लेयर बना लेती है, जस्ट लाइक लेमिनेशन वो कर लेती है। और इसी की वजह से आपने देखा होगा कि हमारी जो स्किन होती है उसमें वाटर, जो ट्रांस एपिडर्मल वाटर लॉस रहता है वो रिटेन रहता है, वो मेंटेन रहता है, और ये रेजुविनेट होता रहता है। इसी वजह से आप स्क्रब यूज करते हैं तो आपके फेस पे, आपकी स्किन पे ग्लो होता है, बिकॉज डेड केराटिनाइज सेल मस्ट बी रिमूव टाइमली। उनका भी एक लाइफ पीरियड, टाइम पीरियड रहता है, उसमें हमें उनको क्या करना होता है, रिमूव करना होता है। नाउ this is the dermis. So dermis is complex, or we can say different structure. It's more thicker, 2 mm around ये thick होता है, और इसी के अंदर इलास्टिन, कोलेजन और लिम्फ ग्लैंड्स होती है, ब्लड वेसल्स होती है, मास्ट सेल होती है, फिब्रोब्लास्ट होते हैं। आपने देखा होगा एटोपिक डर्मेटाइटिस जो इचिंग होती है। तो स्किन के बहुत सारे ऐसे डिसऑर्डर्स होते हैं जो डर्मिस में इरेगुलरिटीज की वजह से, बायोकेमिकल इम्बैलेंसिंग की वजह से, माइक्रोब्स इम्प्रेजेंस की वजह से, मैं आपको बतलाऊंगा कि इसके अंदर आप देखेंगे कि
एक्सक्यूज मी सर,
यस
सर आपकी स्लाइड्स जो है वो चेंज नहीं हो रही है, वहीं पे हैं।
अच्छा, ये भी आपको चेंज नहीं हुआ है
सर? नाउ इट इज विजिबल, पहले वो सेकंड स्लाइड ही शो हो रही थी हमें।
ओहो, नाउ इट्स डर्मिस।
यस सर, यस सर।
इज दिस विजिबल और नॉट?
यस सर।
राइट। तो आप देखिए कि ये अभी डर्मिस वाला ही दिख रहा है ना आपको स्लाइड?
यस सर।
इसमें आपको पूरा स्क्रीन शेयर, फिर मैंने उसको प्रेजेंटेशन मोड में नहीं किया है क्योंकि शायद वो वहां से स्क्रॉल नहीं हो पा रहा था। अभी दिख रहा है?
यस सर, सर शो हो रहा है हमें।
नाउ यू विल सी कि स्किन के अंदर, जो मैं अभी आपको डर्मेटाइटिस के बारे में बता रहा था, स्किन के डिसऑर्डर्स होते हैं. यू विल सरप्राइज दैट आवर स्किन is carrying so many microbes, so many microbial flora. अपना माइक्रोबियल फ्लोरा जो होता है उसको मेंटेन करके चलता है. उसको कॉमनसिल बैक्टीरिया हम बोलते हैं, जो हमारी गट में भी रहते हैं और स्किन में भी रहते हैं. पूरा एक लेयर बनाता है, राइट. सो जनरली वंस द इम्बैलेंसिंग इज अकर, देन स्किन प्रॉब्लम रेज्ड. ये दिख रहा है आपको? चेंज स्लाइड.
Yes, sir. Yes, sir.
नाउ फुल स्क्रीन इज विजिबल और नॉट?
No, sir, it's not full in a normal mode.
In a normal mode.
Yes,
Now
sir.
normal. Okay.
No, sir.
Let it be normal, ना?
Yes, sir.
ये जो जनरल स्किन प्रॉब्लम्स होती है, ये एजेज के साथ साथ आपका एनवायरमेंटल हैजर्ड्स की वजह से आपको देखने को मिलता है. जनरली हमें हमारे फेस के ऊपर आप देखेंगे रफ और क्रिस्पी स्किन जो टेक्सचर नजर आता है. जो आपका ये चिक होते हैं, चिक का जो बोन होता है, चिक बोन जो होती है, वहां आपने देखा होगा जनरली रफ और क्रिस्पी स्किन जो होती है टेक्सचर, वो फॉर्मेशन होने लग जाता है. सम हाउ टाइम बीइंग जो आपके क्रोपोर्ट्स बनते हैं, जो आईलिड्स के साथ साथ क्रोपोर्ट्स बनते हैं, वहां रिंकल्स हमें शो होने लग जाते हैं. सम रिंकल्स raised from this region, right? So these are the reason where the aging reflect, isn't it? Now is this visible? Next slide. General
Yes, sir.
skin
Yes,
problem.
sir. Yes, sir. Yes, sir.
तो जनरल स्किन प्रॉब्लम्स में आप देखेंगे स्पॉट्स बनने लग जाते हैं, रिंकल्स फॉर्मेशन होने लग जाता है, डार्क सर्कल्स होने लग जाते हैं, फिर आपके पिगमेंटेशन. सो देयर इज एक्नी, isn't it? और some other changes, pore formation. स्किन के अंदर जो पोर होते हैं आपके, जो कि स्वेट ग्लैंड के भी हो सकते हैं, सिबम ग्लैंड के भी होते हैं. वंस द पोर बिकम सो लार्जर, तो वो किसका फॉर्म, किस फॉर्म में हमें दिखते हैं? फिर वो एक्नी के फॉर्म में दिखते हैं, क्योंकि वो जो सिबम ग्लैंड्स है, उन सिबम ग्लैंड के अंदर हमारे जो एक्नी वल्गेरिस जो बैक्टीरियाज होते हैं, जो माइक्रोब्स होते हैं, वो उसके अंदर पेनिट्रेट कर जाते हैं सिबम के साथ, सिबम ग्लैंड में, एंड दे मेक द कमेडोज. और वो कमेडोज बना लेते हैं, ब्लैक हेड्स बना लेते हैं, उसके अंदर पस फॉर्मेशन हो जाता है और वो पिंपल्स जो है ब्लास्ट होने लग जाते हैं. So there is different ways. I think some background voice is.
So now it's mute.
So there is so many skin disorder is there, and how we can treat the such kind of disorder. जैसा कि आप जानते हैं, जैसे ही आप फिजिशियन या डर्मेटोलॉजिस्ट के पास जाते हैं, वो आपको कुछ ना कुछ ऑइंटमेंट देते हैं, आइदर क्रीम फॉर्म में या ऑइंटमेंट या लोशन. तो उसमें जनरली होता क्या है, आइदर एंटीहिस्टामिनिक्स होंगे या आपके सम एंटीबायोटिक्स होंगे. नाउ यू विल सी देयर इज ओनली द टू रीजन वी कैन स्पेकुलेट. द मेजर रीजन इज डर्मिस, क्योंकि डर्मिस के अंदर ही बायोकेमिकल चेंजेस हो रहे हैं. इस वजह से आपको हिस्टामिन वहां रिलीज हो रहा है, तो आपके अंदर एलर्जी हो रही है, देन वो आपको एंटीहिस्टामिनिक देंगे. या एपिडर्मिस के अंदर आपके जो सरफेशियल चेंजेस जो हो रहे हैं, उसके लिए इन्फेक्शन हो रहा है, तो वो आपको एंटीबायोटिक्स दे रहे हैं माइक्रोब्स के लिए. इज इट? नाउ beyond such vitamins or steroids or antibiotics or antihistaminic drug, there is several other ways where you can prepare the formulation. You can utilize that natural resources. So beyond the medicine, there is greater opportunity for the holistic treatment, right? जैसा कि हम घर के अंदर देखते हैं, आप यूज करते भी आए हैं, और काफी बिलीवेबल और रिजल्ट उनके जो है परफेक्ट रिजल्ट आपको देखने को मिलेंगे. और ये आज से नहीं, ये ट्रेडिशन तो हमें बिफोर द बीसी चल रहा है, थाउजेंड्स ऑफ ईयर्स से चल रहा है. आपने पुराने, अगर आप एपिक्स को देखेंगे, देयर इज रेफरेंसेस आर कोड, कॉस्मेटिक से रिलेटेड रेफरेंसेस कोड किए हुए हैं. आपने देखा होगा माता पार्वती, गणपति की जो स्थापना हुई थी, गणपति की जो उत्पत्ति हुई थी, वो भी माता पार्वती के मेल से ही, जो उन्होंने उपटन लगाया था, उस मेल से ही उन्होंने एक स्टेचू बनाया था गणपति का, और उन्हीं के अंदर वो जीवित हुए थे. तो आई मीन टू से कि ये रेफरेंस जो है स्किन कॉस्मेटिक के, ये आज से नहीं, हम पुराने समय से करते आ रहे हैं, लेकिन कॉस्मेटिक टर्म जो है ये नया कोइंड किया हुआ टर्म है। लेकिन अगर आप नेचुरल वे में अपनी स्किन को ट्रीट करते हैं या उसकी केयर करते हैं, देन यू कैन सी द गुड स्किन रिजल्ट, राइट? नाउ you can see how we can use and what manner, what will be the mode। अलग अलग मोड में आप इनको यूज कर सकते हैं। आप देखिए द रीजन, अगर हम रीजन को देखें तो स्किन का जो सबसे पहले हम यूज करते हैं कॉस्मेटिक, वो सिर्फ एजिंग और रिंकल्स और हाइपर पिगमेंटेशन को रोकने के लिए हम करते हैं। आप देखिए कि अभी नया रिसर्च जो है वो टॉक्सिक प्रोटीन्स जो हैं वो एक नया सॉल्यूशन बताया जा रहा है। इट इज स्पेकुलेटेड research, we can say hypothesis that the toxic proteins is new solution for the aging skin. And this is proven because our skin is aged due to only due to the collagen, राइट? कोलेजन फाइबर्स का लूजनिंग होना और लॉस ऑफ वॉल्यूम, यानी कि वो थिन हो जाएगा, उसका वॉल्यूम जो है वो कम हो जाएगा। तो लेस वॉल्यूम होने की वजह से ट्रांसपेरेंट और क्रिस्पी स्किन जो है वो अपीयर होता है। अनदर, वंस द लाइनिंग और लाइनिंग ऑफ द कोलेजन विल बिकम रिड्यूस और थिनिंग, once the skin become crispy, it means there is less firmness। तो स्किन का जो फर्मनेस है, उसकी जो टोनिंग है वो आपकी कम हो जाएगी। वंस द टोनिंग विल रिड्यूस, देन इट विल बिकम रिंकल्स, रिंकल्स आपके रिफ्लेक्ट होना स्टार्ट कर देंगे। देन वंस द रिंकल्स अपीयर, देन इट रिड्यूसेस द इलास्टिसिटी। तो इलास्टिसिटी स्किन का फिर आपका कम होना शुरू हो जाता है और जॉवलिन और नेक जो एरिया है वहां आपके रिंकल्स लाइनिंग्स स्टार्ट हो जाती है। सो टू मेक योर स्किन यूथफुल, फर्मर, यू कैन यूज अ प्योर ऑर्गेनिक काइंड ऑफ कॉस्मेटिक फॉर्मुलेशंस। आप देखिए बड़े अच्छे अच्छे जो फॉर्मुलेशंस हैं वो मार्केट में हैं, लेकिन किन्हीं बहुत कम मीन्स 0.1% फॉर्मुलेशंस आर बिकमिंग tried for the scientific research। मार्केट में राइट और लेफ्ट सो मेनी फॉर्मुलेशंस आर सेलिंग आउट, बहुत सारी कंपनीज हैं। लेकिन अगर आप नेचुरल वे में ही आप देखेंगे, देन यू कैन फील दैट these natural formulations are far and far better। some new फॉर्मुलेशंस जो रिसर्च प्रोडक्ट हैं वो मार्केट में आ रहे हैं, कोलेजन बेस्ड फॉर्मुलेशन मार्केट में आ रहे हैं। उन फॉर्मुलेशंस को लेके जो हमने रिसर्च किया वो मैं आपके साथ शेयर करना चाहता हूं। हाउ वी कैन प्रूव अवर फॉर्मुलेशन, हाउ वी प्रूव्ड, व्हाट वर द स्टडी कंक्लूजन, मैं आपको बताना चाह रहा हूं। जो कुछ फॉर्मुलेशंस हमने बनाए थे तो उसको हमने क्यूटोमीटर जो होता है उससे हमने स्टार्ट किया था। क्यूटोमीटर से ये एक डिवाइस होता है, द कॉस्ट इज वेरी हाई ऑफ दिस डिवाइस, बट दिस डिवाइस आफ्टर द एप्लीकेशन ऑफ 2 वीक द फॉर्मुलेशन, this cutometer was used or placed on the skin, so it determined the elasticity at the multiple level, and it determined the skin elasticity as well as viscoelastic property of the skin। And in this particular cutometer, the probe is measured the skin elasticity by its ability to return its previous state। आप ये देख रहे हैं, इसमें आपको दिख रहा होगा ये जो प्रोब है, इस प्रोब को हमने दिस पिंक एरिया इज द स्किन, इस स्किन के ऊपर इस प्रोब को हमने प्लेस किया। देन देयर इज सर्टेनली द वैक्यूम इज क्रिएटेड, तो वैक्यूम यहां से सक करेगा स्किन को। अ पर्टिकुलर पास्कल प्रेशर इज created in this particular probe, and how much skin is displaced the volume, right? So that is a arbitrary unit is noted down in form of the elasticity। So this is the function of the functioning of the cutometer। It is measured specifically for the skin elasticity। Now what are the skin elasticity? So in a detail if we go, it's a negative pressure, right? Negative pressure drawn into the skin aperture of the probe, and inside there is non-contact optical मेजरिंग सिस्टम इज देयर, बड़ा ऑप्टिकल जो है इसके अंदर प्रिज्म यूज किया जाता है, दैट इज वेरी सेंसिटिव। वो माइक्रोमीटर्स जो लेंथ होती है और विड्थ होती है उसको भी काउंट करता है, उसको भी मेजर करता है, उसको भी सेंस करता है। सो द रेसिस्टेंस ऑफ द स्किन टू द बीइंग सक्ड इनटू द अपर्चर इज डिटरमाइंड बाय इट्स फर्मनेस। स्किन जितना फर्म होगा, फर्म मींस टाइटनेस, आपने देखा होगा जो ड्रम्स जो होते हैं, ड्रम जो हम यूज करते थे पहले, वो स्किन के बने हुए होते थे, एनिमल स्किन के बने हुए होते थे वो ड्रम्स। तो उन ड्रम का फर्मनेस कितना होता था कि जब आप उस ड्रम को ड्रम स्टिक से हिट करते थे तब भी वो जो ड्रम होता था वो कभी भी रप्चर नहीं होती थी उस स्किन पे। यानी कि आप देखिए उस नेचुरल स्किन का फर्मनेस कितना होता था। सो दैट काइंड ऑफ फर्मनेस अवर स्किन आल्सो पजेसेस। लेकिन हम उसको क्या करते हैं, उस स्किन के फर्मनेस को खत्म कर देते हैं जो मार्केट में डिफरेंट काइंड ऑफ जो आपके फॉर्मुलेशन है उनको यूज करके। नाउ यू विल सी द डिफरेंट क्वालिटी, और वी कैन से इट्स नॉट ओनली द क्वालिटी, इट इज प्रॉपर्टी ऑफ द स्किन, दैट इज स्किन हाइड्रेशन। स्किन के अंदर रिंकल्स क्यों आते हैं, स्किन के अंदर इलास्टिसिटी कम क्यों होती है? द मेजर रीजन इज द हाइड्रेशन, द लॉस ऑफ हाइड्रेशन। आप देखिए स्किन का हाइड्रेशन अगर कम हो गया, स्किन आपकी डिहाइड्रेट हो गई, एंड देयर इज सेवरल रीजन। आप कोई सा भी सोप यूज करते हैं, टॉयलेट बाथ सोप यूज करते हैं, उसमें भी बहुत सारे हेजार्डस केमिकल्स होते हैं जो आपकी स्किन को हैरेश करते हैं। डिटर्जेंट्स होते हैं, आपकी स्किन की ओक्लूसिव जो लेयर होती है उसको खत्म कर देते हैं, उसके डिहाइड्रेशन को बढ़ा देते हैं। और उसकी आपने देखा होगा जो आप टॉयलेट सोप यूज करते हैं, जो इस बाथिंग सोप यूज करते हैं, उसके अंदर NMF लिखा रहता है, दैट इज नेचुरल मॉइस्चराइजिंग फैक्टर। डव के अंदर बताते हैं सबसे ज्यादा होता है। लेकिन क्रीम सोप्स जो आते हैं आजकल, नेचुरल बेस्ड सोप जो आ रहे हैं, कोकम बटर बेस्ड सोप जो आ रहे हैं, उनके अंदर ये जो नेचुरल मॉइस्चराइजिंग फैक्टर है ये काफी हद तक मेंटेन रहता है। नाउ यू विल सी द रिप्रोड्यूसिबिलिटी एंड एक्यूरेसी बाय कॉर्नियोमीटर। ये जो कॉर्नियोमीटर होता है, ये कॉर्नियोमीटर आपकी स्किन की हाइड्रेशन प्रॉपर्टी को मेंटेन करता है, बिकॉज स्किन इज आल्सो हैविंग इलेक्ट्रिकल प्रॉपर्टी। स्किन का अपना इलेक्ट्रिकल प्रॉपर्टी भी होता है। केमिकल प्रॉपर्टी तो मैंने आपको बताया था, पीएच से आप समझ सकते हैं। स्किन का इलेक्ट्रिकल प्रॉपर्टी भी होता है। स्किन अगर हाइड्रेटेड है तो इसकी कंडक्टिविटी काफी होती है, तभी तो हम स्किन में करंट को महसूस करते हैं। So milliampere current is passes through the skin, and there is two probe are placed at the same region, and the passage of the current is measured. So that is through the corniometer. So it Ohm's law, आपको if you recollect the memory or fundamental principle of the Ohm's law, there is resistance is inversely proportional to the conductivity. राइट? तो कंडक्टिविटी ज्यादा होगी तो रेसिस्टेंस आपका बढ़ जाएगा, यानी मॉइस्चर ज्यादा होगा तो स्किन जो है आपका वो मॉइस्ट रहेगा, अगर उसके अंदर वाटर कंटेंट ज्यादा है तो। राइट? अगर वाटर कंटेंट कम है तो धीरे धीरे आपका रेसिस्टेंस बढ़ेगा, रेसिस्टेंस मींस ड्राइनेस बढ़ेगी। और मॉइस्चर आपका है, वाटर कंटेंट ज्यादा है, यानी कि वो मॉइस्ट है स्किन आपकी। और जो मॉइस्ट स्किन होती है वो हाइड्रेटेड स्किन जो होती है वो इमोलिएंट स्किन होती है और शफलिंग स्किन होती है। और उसी स्किन के अंदर अगर कोलेजन और इलास्टिन फाइबर्स का बैलेंस रहे तो वो स्किन सबसे ज्यादा ग्लो करने वाला स्किन कंसीडर किया जाता है। तो नाउ नेक्स्ट वन इज द मॉइस्चर कंटेंट। मॉइस्चर कंटेंट जो आपका होगा वो 10 to 20 माइक्रोमीटर की जो लेयर्स होती है आपकी स्किन, जो आपकी कोर्नियम जो होती है, स्ट्रेटम कोर्नियम, उसको इंश्योर करता है दैट डीपर लेयर डू नॉट इन्फ्लुएंस द मेजरमेंट। तो ये जो है मॉइस्चर कंटेंट, ये हम फ्रिंजेज फील्ड सेंसर्स होते हैं, फ्रिंजेज फील्ड सेंसर से हम फील्ड कैपेसिटेंस जो है उसका डाइलेक्ट्रिक कांस्टेंट ऑफ स्किन उसको मेजर करते हैं। और उससे आपको जो भी अच्छे रिसर्च इंस्टीट्यूशंस हैं, जो रिसर्च बेस्ड कॉस्मेटिक शॉप्स जो होती है, आउटलेट्स जो होते हैं, वो आपकी स्किन का पहले टेस्ट करते हैं, and after testing your skin they evaluate the parameters, then they will suggest what kind of cosmetic skin dermatology preparation is required for your skin. Now skin surface water loss. So how much water loss is being precipitated with your skin, it is also determined by tevameter. So this tevameter is used to determine the superficial water loss, TWL जिसको हम बोलते हैं ट्रांस एपिडर्मल वाटर लॉस। It's water that passes through the skin, evaporate from the surface in order to outer cell layers. Now you will see the next very emerging technology right now in USA. When I was there, I saw this kind of technology. It's a dermatop technology. Skin टोपोग्राफी जो है उसको आपको देखा जाता है. स्किन टोपोग्राफी में आपके रिंकल्स को देखा जाता है, कोलेजन बंडल के अरेंजमेंट को देखा जाता है, एंड हाउ मच पोर साइज इज देयर, एंड दे स्पेकुलेट कि आपका आने वाला स्किन स्ट्रक्चर कैसा होगा, उसका टेक्सचर कैसा होगा, वो आपको एक शीट बना के आपको पहले से ही प्रोवाइड करा देते हैं कि आपके लिप्स में सेगिंग कितना होगा, चिक्स में नेजल फोल्ड्स कैसे होंगे, ग्लेबिला कैसे होंगे, राइट. सो accordingly they will prescribe the formulation. So fine lines, wrinkles and folds, जो folds होंगे उनको भी वो measure करेंगे. So this is called the dermatope, and this dermatope is widely used right now in the research of dermatology. Then this is the mexameter. Mexameter is generally considered to measure the skin color. आपने देखा होगा हाइपर पिगमेंटेशन की वजह से जो है स्किन में डार्क सर्कल्स हो जाते हैं, और उन डार्क सर्कल्स को अगर इनिशियली नहीं रोका जाए तो वो पिगमेंटेशन टैनिंग में कन्वर्ट हो जाता है और स्किन आपकी डार्क जो है डार्कनिंग की तरफ जो उसका बैंड है वो बढ़ने लग जाता है. सो इन दिस पर्टिकुलर मैक्सामीटर द प्रोब इज of this mexameter emits the three wavelength of the light that chosen the corresponding to the different absorption rate of the melanin and hemoglobin, because melanin jo hai that is the factor which consider the color of your skin. Now glossimeter, now you will see this glossimeter is generally used for the skin glow as well as for the hair luster, क्योंकि जब हम कॉस्मेटिक में शैम्पू जो है सेकंड मोस्ट फॉर्मुलेशन हैं जो कि वाइडली यूज किया जाता है. सो दिस ग्लोसीमीटर इज यूज फॉर द हेयर प्रिपरेशन, स्पेसिफिकली फॉर द हेयर टोनिंग एंड एज वेल एज फॉर द कंडीशनिंग और शैम्पूइंग. सो after the shampooing or after the conditioning, how much your hair is nourished, how much hair is healthy, उसका मेजरमेंट किया जाता है इसके रिफ्लेक्शन से. सो ग्लोसीमीटर विल मेजर द रिफ्लेक्शन रिफ्लेक्टिंग लाइट in form of the lux. Lux में उसको measure किया जाता है. Lux is the unit. So measurement of the glow is very important in the efficacy testing of the skin care as well as hair care decorative products. In skin and hair are supposed to show the natural luminous glows without appearing oily. राइट, नेचुरल ग्लो होना चाहिए, ऑयल के साथ नहीं. ऑयल का भी किया जाता है, दैट इज आफ्टर द बेस लाइन ट्रीटमेंट. एंड आल्सो फॉर द नेल्स, ये जो ग्लोसीमीटर है ये नेल्स के लिए भी यूज किया जाता है, एंड इट्स शोइंग द वेरी इंपॉर्टेंट पैरामीटर in the cosmetic. Now skin pH, so skin pH is also one of the factor which are being considered in the cosmetic. Now you will see the advantage because the modern and high quality electronic probe होता है ये स्किन पीएच मीटर. और सबसे पहले वैसे देखा जाए तो स्किन पीएच मीटर से स्किन का ही मेजरमेंट, पीएच का ही मेजरमेंट होना चाहिए. वेयर द आयंस, आपने जो पीएच होता है हमारा वो H प्लस आयन के ऊपर डिपेंड करता है, माइग्रेशन ऑफ द H प्लस आयन. सो जब हमें वो मालूम होता है कि हमारी स्किन का पीएच कितना है, देन वी कैन डिसाइड कि व्हाट काइंड ऑफ फॉर्मुलेशन इज टू बी यूज्ड फॉर आवर स्किन. सो this is the pH meter. Now skin thermal meter is also used for this particular purpose, and it's allow the quick measurement and continuous measurement over the longer period of time. Now you will see the ideal comparison. ये स्किन का जो थर्मोमीटर है ये आपका अलग अलग जो स्किन सरफेस एरिया है वहां का टेम्परेचर हमें नोट करते हैं, ताकि हमें ये मालूम चले कि वहां ब्लड कैपिलरी के अंदर सर्कुलेशन कितना हो रहा है. कि जितना ज्यादा सर्कुलेशन होता है उससे टेम्परेचर को हम एज्यूम कर सकते हैं कि उसकी रेट ऑफ सर्कुलेशन कितना है, और रेट ऑफ सर्कुलेशन के हिसाब से हमें मालूम चलता है कि स्किन का थिकनेस वहां कितना है, और उसी के अकॉर्डिंग हम कोलेजन बंडल्स की रेट को फाइंड आउट करते हैं और फॉर्मुलेशन तैयार करते हैं. नाउ फ्रिक्शन मीटर, दिस इज आल्सो समहाउ कंसर्न विद द ग्लोसीमीटर. फ्रिक्शन मीटर इज यूज्ड फॉर द इमोलिएंट, राइट. इमोलिएंट प्रॉपर्टी के लिए इसको यूज किया जाता है कि स्किन के अंदर जब आप इमोलिएंट यूज कर रहे हैं, क्रीम यूज कर रहे हैं, तो आपकी स्किन के अंदर सपलनेस कितनी है, फ्रिक्शन कितना है, स्लिपरीनेस कितना आया है. So that can be used by the friction meter. There is rotational speed friction meter is there, so it allowed for the measurement over a longer period of time, and this probe head can easily be cleaned as well as measure the rate of friction. Now indentiometer is generally used for the sagging, स्किन सैगिंग, यानी कि स्किन की जो सैगिंग प्रॉपर्टी है उसके अंदर टफनेस कितनी है उसको मेजर करने के लिए हम इसको सैगिंग मीटर को आप देख रहे होंगे। इंडेंशियोमीटर के अंदर आप ये जो स्किन है, ये प्रोब ज्यादा प्रेशर आ रहा है, इसमें कम प्रेशर है, इसमें और कम प्रेशर है। ये आर्बिट्री यूनिट जो है एक नॉर्मल स्किन के अंदर देखी जाती है, देन न्यूली बोर्न चाइल्ड के अंदर देखी जाती है, टीनेजर्स के अंदर देखी जाती है, देन एज्ड पीपल्स के अंदर देखी जाती है। देन अप्लाई द फॉर्मुलेशंस, और उस फॉर्मुलेशन से इंडेंशियोमीटर के अंदर आप देखेंगे कि फर्मनेस कितना बड़ा है स्किन का, वो हम उसको लेते हैं, और वो फर्मनेस जो होता है वो आपका एंटी एजिंग फैक्टर को शो करता है। ना विजुअलाइन क्राउ फिट्स, ये जो है ये विजुअलाइन क्राउ फिट्स जो टेक्निक है, दिस इज इंटेंडेड फॉर द मेजरमेंट ऑफ द रिंकल्स ऑन द फेस, उसके लिए इसको हम जनरली यूज करते हैं। Now visopore, this is also meant for the pore size of the sebum as well as sweat gland, so this is also used in case of स्कल्प। जब स्कल्प के अंदर डेंड्रफ बहुत ज्यादा हो जाती है, आपने देखा होगा कि स्कल्प के अंदर और स्किन के अंदर भी डेंड्रफ होती है। नाउ वेरी इमर्जिंग फील्ड इन दिस पर्टिकुलर एंटी डेंड्रफ इज द प्रोबायोटिक्स। नाउ वी आर थिंकिंग फॉर द प्रोबायोटिक्स। हम प्रोबायोटिक्स, आप देखिए यूएस के अंदर काफी अच्छा रिसर्च हुआ, प्रोबायोटिक्स ही अप्लाई करके उन्होंने स्किन की प्रॉब्लम्स को इंप्रूव किया, स्किन की जो हेल्थ थी उसको इंप्रूव किया, प्रॉब्लम्स को कम किया। द आइडिया बिहाइंड दिस इज दैट टू हेल्प द मेंटेन the beneficial balance of the microbes, helping to reduce the symptoms of the skin condition. And you will surprise, according to 2019 and a study surveys that the composition of the skin microbiomes in the healthy skin differ from the composition of the microbiomes in the diseased skin, राइट। और अभी जो है डिसबायोसिस, हम इसको बोल रहे हैं कि डिसबायोसिस स्किन के अंदर हो जाता है, दैट इज ड्यू टू द वर्सनिंग कंडीशन ऑफ द स्किन। सो देयर इज सो मेनी माइक्रोब्स, द रोल ऑफ माइक्रोब्स इज एसोसिएटेड। काफी सारे माइक्रोब्स की एक वेराइटी होती है जो हमारी स्किन के प्लेथोरा को मेंटेन करती है, बट जब स्किन के अंदर कहीं भी इम्बैलेंस होता है डर्मिस के अंदर, तो वो माइक्रोब्स इन्वेड कर जाते हैं, इन्फेक्शन का कोज करते हैं, राइट। सो माइक्रोबायोम्स बैलेंस इज वेरी मच रिक्वायर्ड, एंड दिस इज द हाई थ्रेशोल्ड रिसर्च domain also. So in the last I can say that we should not use random cosmetic formulations. Cosmetic formulations or dermal formulations must be verified from the research laboratory. बहुत कम formulations हैं जो research laboratory से पास होके market में आते हैं, and we should also having some knowledge, we must do ourselves educate. हमें educate करना चाहिए अपने आप के बारे में, अपनी skin के बारे में, हमारी skin कैसी है, then we can use that kind of formulation and don't use unreliable marketed formulations. There is lot of scope in this particular dermal and cosmetic research right now. And now if any questions regarding this one, please you ask the question. Any queries if you are having?
Yes. Excuse
Thank
me,
you.
Pratyansha ma'am. I
Yes.
have one query. Good morning, sir. I am Aman Patyal from the Department of Pharmacy. Sir, I have one question. You have explained one cutometer apparatus.
Yes.
Is this cutometer works on the same principle as massages do?
Yes. Massage is not only means cutometer is not concerned with the massage. Massage is concerned with the elasticity. जो आपने corneometer देखा उसमें भी moisture के उसमें भी काम आता है, वो भी आप कर सकते हैं, वो भी उसको concern means आप उसको correlate कर सकते हैं.
Okay, sir. Okay. And sir, I have one more question. In today's life, the cosmetologist will opt that laser treatment.
Laser treatment, yes.
Yes, sir. तो सर ये लेजर ट्रीटमेंट्स की ऑलरेडी ही अपनी रेडिएशंस होंगी, तो ये हार्मफुल
Laser
होगा या?
treatment is just like the abrasion of the uppermost layer, right? अब आप देखिए लेजर ट्रीटमेंट जनरली किसके लिए किया जाता है? हाइपर पिगमेंटेशन के लिए, ब्लिस्टर्स के लिए, उनके लेजर ट्रीटमेंट किया जाता है. सो आई ऑलरेडी कोडेड दैट this slide, right? Loss of
Oh,
volume of thinning of the collagen. अगर कोलेजन की थिनिंग होगी तो आपकी जो ऊपर की जो क्यूटीन लेयर है, जो केराटिन लेयर है, देयर इज हंड्रेड्स ऑफ केराटिन्स आर लेयरिंग जस्ट लाइक अ रूप टाइल मैनर. जो केराटिन्स होती है रूप टाइल मैनर में इस टाइप से चिपकी रहती है, राइट? एंड दैट केराटिन्स आर अरेंज्ड वेल. आप देखिए वो इतने अच्छे से चिपकी रहती है. किससे चिपकी रहती है? द एडहेरिंग सॉल्यूशन इज नेचुरल, दैट इज सीबम, राइट? सो सीबम इज
okay.
कमिंग फ्रॉम वेयर? It's coming oozing from the dermis layer.
Glenn.
So if your dermis is healthy, then all the keratins will arrange in fine manner. If it is disarranged, means there is some irregularity in the biochemistry of our dermis. Is it?
Oh, okay, sir. Thank you. Thank you so much.
Thank you, ma'am. So we have other query. The first question is, do you recommend use of same product regularly throughout the year, or we should use different products?
No, ma'am. As I already coded that skin transepidermal water loss is different. In winter, there is different water loss. In summer, there is different water loss. In winter, there is different oil content. In summer, there is different oil content. So all depends on the weather. समर के लिए अलग फॉर्मुलेशंस होते हैं, विंटर के लिए अलग होते हैं, स्प्रिंग के लिए अलग होते हैं. एंड फॉर्मुलेशंस आर चेंजिंग डे बाय मींस मंथ टू मंथ. आप दिसंबर विंटर में भी जो है अलग अलग फॉर्मुलेशंस यूज कर सकते हैं. हाईली जो लो टेम्परेचर रीजन होते हैं वहां के लिए हाई फैट कंटेंट और कोकम बटर बेस्ड फॉर्मुलेशंस होते हैं, उनको यूज किया जाता है. और इनिशियली जो नवंबर मंथ होता है, जो हमारा टेम्परेचर जिसमें 30 और 28 का जो टेम्परेचर रहता है, उसमें अलग फॉर्मुलेशंस को हम रिकमेंड करते हैं. एंड समर इज एंटायरली डिफरेंट. समर के लिए फॉर्मुलेशन से अलग होते हैं.
Okay, sir. So the next question is, how we so it's your device that it is a economically high cost, but economically weak people, how can they use that
We
instrument?
cannot, ma'am, that's why I'm telling you, we cannot use in our home, na. This device is not meant for, because the one device cost is around 3 to 4 lakh. So it's not possible. I just want to submit my submission and means what I am giving you. I am telling you कि हम जो फॉर्मुलेशन यूज करें वो ऐसे हो कि कम से कम लैब पास्ड हो, क्योंकि हम इंडिया के अंदर वो फॉर्मुलेशन यूज करते हैं जहां ये रिसर्च नहीं किया जाता है. सो बेटर ये है कि हम सोच समझ के फॉर्मुलेशंस को यूज करें, और अदरवाइज नेचुरल फॉर्मुलेशंस को ही यूज किया जाए, ज्यादा बेटर.
Okay, sir. So the third question is, how would we handle the situation during the burn?
Ma'am, if we talk about the natural method, there is a chilled water or cool water. Immediately you just expose the burned skin through the cool water or cold water. Cold water से आप स्पॉन्जिंग कर दीजिए और उसके बाद तुरंत प्राइमरी ट्रीटमेंट. और सबसे बेस्ट जो है वो, अगर आपके घर, हर घर के अंदर एलोवेरा का प्लांट होता है, उस एलोवेरा को आप तुरंत जो है स्क्रैप करके, उसमें येलो पोर्शन होता है वो एलोइडीन होता है, उसको रिमूव कर दीजिए, बिकॉज दैट इज कार्सिनोजेनिक. उसको निकाल के और जो व्हाइट पल्प होता है उसको आप प्लेस कर देंगे, देन इट विल गिव यू मोर देन 50% रिलीफ एट अ टाइम.
Thank you so much, sir,
Thank
for
you.
sharing your deep knowledge about the modern biological tool, cosmetic dermal formulation with us. Your session was full of passion and valuable knowledge. Thank you once again, sir. Now, this is an announcement for all the participants that feedback form will be shared very soon. E-certificate will be provided to those students who will fill the feedback form. Now, we have a second speaker. Now we have a second speaker, Professor Dr. Girish Kumar Gupta. He is Director Research and Development, Sri Sai Group of Institute, Badani, Pathankot. Sir has 14 years of experience in academic and 2-year industrial experience. He is honored with Pavilion Peer Review Award, App West Researcher Award, and Young Scientist Award. He is selected and nominated for Top 100 Scientist. Sir is also nominated for Rising Star of the Year and Future Pharmacist of the Year. He has published more than 100 publications at national and international level. He has awarded with 4 patents, 2 copyright. He has published, edit books, and Sir is also editor and guest editor of number of journal. He is a lifetime member of different organizations. He has traveled to different nations to win numerous honors and recognized from professional association. Now, I would like to call Professor Dr. Girish Kumar Gupta to deliver his talk on the topic chemical and biological importance of natural products. Over to you, sir.
Hello, am I audible?
Yes, sir.
Thank you. Uh, Thank you, Dr. Prachanta Sharma, for overwhelming welcome address. I am very much thankful for the management as well as the organizer to giving me this opportunity to present my views about the hot topic of the era, that is natural products. I am going to share my slide. Please let me know whether you are able to see.
Okay, sir.
I hope screen is visible now.
Yes, sir.
Yeah. So all right. Here I am going to present a very hot topic of the era, that is natural products components. And as all of you know, that natural products is a chemical component or compound that has been produced by some living organism. Okay, living organism can be anything. It is about the natural bioproducts or natural biological species or plants, or they may be prepared chemically as well. So, by definition, if we say that natural products, what are these? They can be referred to as cosmetics, dietary supplements, and food products from the natural sources without adding any type of artificial ingredients. Okay, so these type of components are very much helpful or interrelated with two basic branches of the science. One is organic chemistry as well as medicinal chemistry. So why we study the natural products? Basically because their sources are most complex and they are very fascinating chemical structures which attract the interest of the pharmaceutical products as well as
Hello, excuse me, sir.
pharmaceutics. Hello.
Hello, excuse me, sir. Sir,
Yes.
only first slide is visible. Third day nation symposium.
Just wait. What about now?
Yes, sir. It's visible, huh?
Okay. Okay. So please let me know when there will be some interruption. Please, you are welcome to know that whether slides are moving or not. All right. So what are the natural products basically, and why we will need to study this process or these science? Because of their complex nature as well as the fascinating chemical structures. Okay. And also they have biological diversity. They have numerous expressions of genome. They have multiple properties in a single component. Okay, so and overall they are the major part of economy as well as the ingredients of livelihood sources, agriculture products, food products, pharmaceutical products, chemical industrial products likewise. So overall in this slide you can have an overview that how the natural products can be related with the ecology of the environment, and
Excuse me,
you
sir.
can see. Yes,
सर प्लीज आप स्लाइड शो कर दो इस चीज का.
it is already may I know from where it can be done?
सर जहाँ पे अभी आप अपना माउस लेके गए थे वहीं पे जाना है वापस. So, वहीं पे क्लिक कर दो. सर और नीचे सर क्लिक है.
Is it visible now?
No, sir. So you can continue with this.
That's why I have make the previous view, because you are saying that the slides are not moving.
Yes,
However,
sir.
I have shared the same. If you can wait, I will again share the slides.
No, sir. It's okay. It's okay. So you can continue.
Okay.
Okay, sir.
So you can see that they can be the part of animals, insects, various types of microorganisms like bacteria, fungi, and there is an equilibrium, symbiotic relationship, as well as pheromonic relationship, chemotaxis communication. So all these things are related or interrelated with the natural products, which can affect our environment directly and indirectly. This is a general isolation strategies which we academicians or pharmaceutical industrial people will do. That is, they should take a raw plant, they will do solvent extraction process, they will extract a crude extract, as well as they can do the fractionation to find simplify the complex extract, and then through the various chromatographic techniques or purification techniques they can identify or simplify the complex component to the pure component, and then by the help of the structural elucidation techniques, either they are UV, NMR, IR, LCMS, HCMS, that's why they can help in the formation or structure determination. So I'm not going to the general process that how it can be isolated in pharmaceuticals, but because my major focus is on the their biological and chemical component. So you can see that basically the natural products have carbon skeleton. So this is a table where we classify various types of natural products in different ways on the basis of the carbon atom they have. So if they have C5 carbon atoms, they can be categorized into isoprenoids. Okay, if they have C6 carbon atom system, they can be simple phenols or phenolic derivatives. If they have more than six or fused system, they can be categorized as cinnamic acid derivatives, naphthoquinones, terpenes, xanthones. If they have C6, C2, C6 systems, they can be categorized in anthraquinones. If they have C6, C3, C6 system, they can be categorized into the category of flavonoids. If they have different, so you can see on the slide that there are different type of carbon chain arrangements. They can be lead to the different type of natural products. They can be sesquiterpenoids, lignans, diterpenes, complex flavonoids, bioflavonoids, triterpenoids, carotenoids, lignins, tannins, and sometimes they can be alkaloids, which can be called glycosides. So you can see from these type of classification, there is a huge class of natural products available or come to existence in the literature. So there are other several ways to categorize them beyond this carbon skeleton system, that is like flavonoids. We can be called or categorize these types of component as oxygenated derivatives of the which have aromatic ring system. Okay, we can also categorize from their genus of the plant, like morphine and codeine. They can be extracted from opium alkaloids. So this is the another basis of classification, or we can also classify them on the basis of some physiological effect what they exist. They may be antimicrobials, they may be cardiotonics, they may be analgesic, they may be antibiotic drugs. Okay, also we can also categorize these types of natural products by the similarity or roots, how they can be synthesized from the microorganisms, how they can be biosynthesized. All right. So in this slide, I have made an attempt that how natural product chemicals or chemistry or products can affect or interrelate with other types of branches. Either it has a pharmaceutical science, is biochemistry, it is an entomology, combinatorial chemistry, pure organic chemistry, microbiology, biotechnology, molecular biology, etc. So, as I said, they can be available from various types of natural sources. So one of the sources is microorganisms like bacteria and fungus. So antibiotics are the group of class which are available as a natural product sourced from the microorganisms. Marine microorganisms, they can also have potential antiviral as well as anti-cancerous activities. And vast type of marine organisms are available in the literature. They have been isolated from. They have also available from higher plants like morphine, quinine, taxols. They can be available from sponges. They can be available from higher animals as well. So this is way that say you can say natural products are widely available in nature. On the major basis, I have focused basically on the secondary metabolites because this is a wide area. It is not so easy task to compile all the things in 1 or 2 hour lecture. So I have focused on some few categories which are widely important and of quite interest if you see in detail, like polyketides and fatty acids, terpenoids and steroids, phenylpropenoids, alkaloidal drugs, amino acids and peptide containing compounds, carbohydrates, and flavonoids. They can also be uh, categorized into, or you can say, enzymes and coenzyme category, purines and pyrimidines and nucleic acid category, steroids and hormonal drug containing components, natural pigment containing components, lipids, antibiotics, and vitamins as well. So all these types of subcategories can be categorized into natural products. So today my area of focus about my special talk is about these four categories, that is terpenes. On that I can basically go with pheromones, alkaloids, important steroids and hormones, and few pigments. So what are terpenes? Terpenes are the most interested carbon skeleton natural products. And basically they can be 2-methylbutane residues which have isoprene type of subunits, which can be either polymerized or you can say arranged in a sequential way, leads to the formation of different type of other derivatives. So you can see isoprene, how it can looks like in the structure. And if we say okay, it is having head and tail portion and how it can be connected, you can say like that. So this type of structural arrangements are available in the terpenes molecules, that is isoprene. So if they classify terpenes into subcategories, you can say if two isoprene units are arranged as we show in our previous slide, you can say it is monoterpene types of components. If three type of isoprene units are having carbon 15 in total, they can be categorized into sesquiterpenes types of moiety. And if they are four in number, they can be categorized into diterpenes and so on, like triterpenes about. So there are few examples. These are available in the literature and have been isolated from the various scientists and labs, like myrcene, geraniol, terpenine, cineol, menthol, menthone, carbone, thuzone. Many of you might be heard these types of component or seen these components on the leaflets as well as the outer, you can say, uh, decorative labels of various natural drugs or food products. Sesquiterpenes examples are farnesol, udinisol, bulgarinin, atracyclinone, caryophyllene, aromadendrin, germachrone, linderine, etc. Some other representative diterpenes are phytol, pymeric acid, quarine, abietic acid, marubin. Some representative triterpenes are lanostane, oilane, lupol. You see, these types of dry terpenes have some steroidal type of arrangement, including isoprene as well. So, isoprene units and terpenes are also available in various types of natural products like limonene. You can see this is the isoprene arrangement which has been demonstrated in the blue color in dark, how the isoprene units can be arranged in the aromatic component. In camphor as well, you can see this type of structure arrangement is present as the isoprene unit. In selenin, you can see there is a structural isoprenyl sequential arrangement that can be called this type of component, as well as guaiacol. Non-isoprene terpenes are also available, which does not have the same type of structural arrangement, but they can be categorized in the terpene category as well as good natural products, that is valerian, Vivitone, aromophenone. They can be the components of various types of cosmetic products. You can see in perfumery industries, these types of agents can be used. So, what is the importance basically? If we categorize, or I have discussed earlier, that what are monoterpenes, sequiterpene, diterpene, triterpenes, the various examples we have seen in our previous slides. So you can see that monoterpenes have basically essential oil types of properties, and they have been used for their antibacterial activity. Sesquiterpenes, they can be found naturally in the insects as well as plants, and they can be used as a defensive agents and pheromones. I can demonstrate few examples which can be of quite interest with a chemist as well as natural product chemist, so they can be do or take the lead molecules for their research to carry out further on. Likewise, diterpenes, they have some antimicrobial as well as anti-inflammatory property. Triterpenes have some type of basic structures, as we seen, that is steroidal type. So they are also very wonderful anti-inflammatory agents. So now you can see that uh, this is the example where we have some isoprene units. They can be found in the component called retinal. So you can see from the slide that it is cis retinal component and trans retinal component, and this molecule is known as chemistry of vision. Basically, due to this component, we have a vision property of our physiological body system that can be activated through brains or nerves. So you can see that how wonderful natural products are. They can be available with all these types of physiological system. So next is pheromones, and most of you might be known that this molecule is able to control your uh, communications or way of showing your love, or you can say attraction to the second communicating agents. Either they are insects or some other biological source. So what is pheromone? Pheromone is basically secreted and excreted as a chemical factors which can be a trigger response of the social members in the same community. So what they can do, they can also act as a ectohormones, and they can control the love life and affect various types of communication aspects in insects. So we have seen this type of arrangement in ants. Most of the time we see how they can be communicate with one to each other out about the search of food, etc. So humans, however, do not seem to be do with the same type of secretions. However, the pheromones type, you can say it is a endostenodione type of steroidal moiety that can be act as a pheromone, and or you can say it is a male sweat that can be able to uh, uh, make an attraction behind the uh, you can say the communication sources. So that can be detected somewhere in very easily in insects, but in humans it can be not detected very easily. We can presume such type of hormonal changes in the individual to individual species that can also affect their behavior as well as the physiology. So likewise, you can see in the slide that serotonin, that is also known as a cigarette beetle, daspirin, that is available from gispy moth, olean olive, it can be available from olive fly. So these types of pheromones or so many others, they have one sex usually from the female part of the insect, which can be used to attract the mates for the breeding. So these are very wonderful agents if available already synthesized form or isolated from the insects, and can be utilized for different type of other drug manufacturing or biological alteration type of uh, research. So you can see that how wonderful these things. Then males may emit the pheromones to convey the information about their species and genotype. And these types of moths, butterflies, beetles, they can detect these types of pheromones of potential mate as far as from the 10 kilometers. Okay, it is just an idea. It may be more than that or less than that. So in some insects, there is a single molecule of sex attachment that can elicit the same type of response. More, that is, they can be useful weapons as well, or you can say biological weapons. Okay, so this has been used to mask the or making the confusion the, you can say, other species, so that they can be attracted towards the different side, and you can be able to protect either your crops or your particular plant species or particular uh, uh, potential agent which have the biological activity. So one of the example is that is cotton ball weevil, and it can reduce the need of conventional chemical insecticide by using these type of pheromones. You can say alarm pheromones. Alarm pheromones can be available from aphids. They can be used to trigger aggression in ants, honeybees, termites. Okay, so what they will do, they emit pheromones when grazed upon and resulting in the tannin production in the neighboring plant. And these tannin makes the plant less appetizing for herbivores. So by this way, they can also make them protect. So trail pheromones next, that can be used for social communication. They have some volatile hydrocarbons that can be excreted and can be detected by the other neighboring species. Royal pheromones, royal pheromones like queen honeybee. So queen honeybee, what it will do, it will attract the worker bees by her pheromones to build her mandibular glands. Okay, so these pheromones can be excreted from that, and they can be a mixture of alcohol or organic acid that can be volatile in nature, and they can be available in the environment to detect their fragrance, and rest of the bees can follow the same what the message the queen bee is conveying to them. So what they will do, they can be used this to workers to feed and groom her. They can inhibit the workers from building the queen cells or rearing other new queens, or inhibiting the ovary development in the workers. So this is the fascinating phenomena that is existed in the environment, and how they can do the communication. So that's all about the pheromones. Now I'm going to discuss about alkaloids. There are other second category of natural products which is widely available in the environment, biodiversically available, but difficult to synthesize. They can exist naturally, but they are difficult to synthesize chemically. However, several research organizations are doing wonderful part of preparing their derivatives. So basically, you can say they have nitrogen containing species, as you've seen in the slide, that is pyrrolidine, piperidine, tropane, pyrrozolidine, indoles, isoquinoline, quinoline. So all these types of heterocycles which have nitrogen, carbon, hydrogen atoms, also some heteroatoms, they are available. They can be oxygen, sulfur, chlorine, halogens, or phosphorus. So all these types of natural products have possess some activities. Here is a list like anti-malarial. So quinine is an alkaloid which have anti-malarial activity. Aphidine is an alkaloid that have anti-asthmatic activity. Harigatonin is a alkaloid that have anti-cancerous property. Vinca alkaloids, they have specifically vincamin. They have vasodilator activity as well as anti-cancer activity. Quinidine is an alkaloid which have anti-arrhythmic activity. Morphine is a drug or natural product which have morphine as an alkaloid, and it has analgesic activity, well known worldwide. Clarithin is an having antibacterial activity. Piperine have anti hyperglycemic activity. So all these types of pharmacological activities are exist in the natural product. How wonderful this natural product is. So other than that, you have psychotropic effect, that is psilocin. You have find some stimulant activities, that is central nervous system stimulant activities. So that can be exist by cocaine, caffeine, nicotine, theobromine, which are available in biological source in our tea, coffee, etc. And also they have toxic effect as well, as that can tubercularin, a very well known toxic agent which have been used in ancient time to kill, or you can say hunting in the animals. Okay, so atropine is a very well known uh, uh, poisonous alkaloid, but can be utilized for a drug in a small quantity or minor quantity. They have some bitter taste as well. So overall, alkaloids can be classified into four subtypes, that is true alkaloids, protoalkaloids, pseudoalkaloids, as well as false alkaloids. So what are true alkaloids? They are derived from the amino acids. They have nitrogen in the heterocyclic ring. The example is atropine. We have just seen the structure. Protoalkaloids, they are derived also from the amino acid, but they do not have nitrogen in the heterocycle ring. However, they have heterocycle ring in the fused form. The drug is ephedrine, a very well-known example. Pseudoalkaloids, they are not basically derived from amino acid, but they have nitrogen in the heterocycle ring, and the best example is caffeine. Okay, so as a that is false alkaloids, they can also non-alkaloidal nature, but they give false positive reaction with the alkaloidal reagent. However, they have not the structural arrangement similar like other true, proto, and pseudo, but they have given the same type of chemical reaction used for the detection of the alkaloids in the reaction mixture. So you can see that these are very good example and categorization. What are the basic functions of these alkaloids? They may act as a protective, as we seen, against the insects, okay, and herbivores, because of their bitterness and toxic property. They are also be waste products in some cases. They are very well known source of nitrogen and can be utilized where the nitrogen deficiencies cases are available. They are also act as a growth regulators in metabolic systems and act as a source of energy as well. So you can see from the slides that how opium looks like, and what are the various types of biological sources components are extracted from that, and that is heroin, codeine, morphine, methadone. So you can see from the structure that they have uh, oxygen as a heteroatom, nitrogen as a heteroatom, cyclic ring as a fused form also available, and these are basically morphine category alkaloids having analgesic activity as well as CNS stimulant activity, respiratory uh, depressing activity as well as. So, what are these types of components can be categorized? They can be categorized into narcotics, alkaloids, and what is this narcotic chemical is? A narcotic is a chemical that dulls the mind or induce sleep or gently numbs all the senses by sequential manner. Okay, so most of the basic category, or you can say prominent category in this alkaloid category is opium. So opium can be available as a poppy, and it can be used beyond than 6,000 year ago, and it has the power to ease the pain. Okay, the morphine is the major component which can be extracted from that as an alkaloid, and it can be in previous time they can be used a word Morpheus. Okay, that the Romans were used for euphoric activities. Okay, so other than that, they have potential analgesic, cough suppressant, as well as psychological responses to induce euphoria. Next is caffeine. Almost all of us have used this natural product in the terms of your coffee as well as tea. So how its structure looks like, you can say caffeine, basic structure, fused ring with nitrogen as an heteroatom, and it have CNS stimulant activity which can restore your alertness. This also in a pure form act as a psychoactive drug, but there will be a limitation for every natural products. If you consume in a higher amount, it can result into the toxic effects. So how the caffeine can have positive and negative effects, you can see it will increase the attention and alertness in case of fatigue. It can also lowers the cardiovascular disorders. It can also the risk lowers the risk of diabetes. It can also increase the metabolic rate, which can helpful in the obese patient to reduce their weight. And what about the negative effects? On contrary, you can see it can lead to addiction. It can lead to anxiety. It can increase the vasoconstriction as well as blood pressure if consumed in a higher amount. It can also reduce the control of your motor movements as well as increase the stimulation of urination. So act as a diuretic. Other than morphine, you can see quinidine type of alkaloids or cinchona plant containing alkaloids, which can be named as quinine, quinidine, chloroquine, mefloquine. They have anti-malarial as well as anti-arrhythmic activity. So that can be helpful agents to maintain your cardiac rhythm. And also these types of molecules have been used from various ancient times, and number of research groups in India as well as abroad, they are moving towards their derivatization as well as semi-synthetic synthesis to produce some one more better component which have less toxic effect. Other natural alkaloids which have indole ring as an heterocycle, they have tryptophan. It is an essential amino acid. Serotonin, it is a wonderful neurotransmitter. Melatonin, it is a hormone. Auxin, it is a plant growth hormone. And other indole alkaloids which are available from the plants, there is a species that's Psycho mexicana, Psilocybe semi-anticia, Rauwolfia serpentina, and what the extract it has been obtained from them is reserpine. And reserpine is a wonderful molecule which can be used in the natural product to control the blood pressure. Next is another third category that is the focus of today's presentation is beta phenylethylamine. So as it has been represented by the structure, beta phenylethylamine, it is an alkaloid and also responsible for falling in love. Okay, so you are surprised to know that this particular component is responsible as a love hormone, or they are the neurotransmitter which are affect your emotions. Okay, so they can affect your warmth, they can affect your affection, they can affect your sexuality, they can affect your feeling of physical energy likewise. Okay, so in short, abbreviated form, it is called P. And that can affect your human psychological behavior associated with particularly falling in love category. According to Walsh hypothesis, when we meet with someone who attract us, the whistle blows. However, it is a theory, but ultimately what happens that a precursor, which is a carboxylic acid derivative, on enzymatic decarboxylation lead to the generation of this molecule, that is P. And due to the help of this monoamine oxidase enzyme, it is further released into phenylacetic acid. So how this biosynthesis can lead to further uh, our biological metabolic process. So particularly this P, it can be released by this phenomena. So Walsh has presumed that when we meet or attract with someone, these hormones can be released from your neuro storage globules. Next is another molecule, that is beta endorphin. So beta endorphin is a very complex molecule. You might be not visible in the slides, but it is a sequential arrangement of 31 amino acids. They can be arranged as I have shown in the slides, that tyrosine, glycine, phenylalanine, okay, methionine, threonine, serine, glucine, likewise. So it is a complex structural arrangement. And what they can be, they have some morphine type of properties, as well as uh, endogenous system that can be act as a natural painkiller. So what these components which arrange in a this sequential amino acid arrangement, they can potentiate the analgesic effect more than 80 times to morphine. So this is already available in our uh, body as a natural component. So endorphin is the morphine that is produced when body feels pain. When in love, it has the same effect as heroin and opium, which can cause the feel happy and joyful psychomotor activities. While we eating fat, we eating chocolates, we eating sugars, they can produce more endorphins. So when they produce in our body, we will feel joy, we will feel good. Okay, and that will ultimately gives our profound relaxation. So that is the structure of morphine. Another important uses of alkaloids, first is as a medicine, that is digoxin. So all of my pharmaceutical colleagues or academicians, they know that digoxin is a wonderful molecule, act as a cardiac glycosides and used for the heart failure properties and maintaining the heart function and maintaining the life. Second is vinca plant. Vinca plant is also a very good anti-cancer drug. The vincristin, vinblastin are the isolated components that are also alkaloidal in nature. So how this plant looks like, you can see from the slide. Next is ephedra, ephedra plant. So the component which has been isolated is ephedrine. So ephedrine also used for the maintaining the blood pressure. Next is codeine. Codeine is act as a cough suppressant. Next, argolin. Argolin is available from the argots, and argot is a group of fungi, you can say, and that can be used to relieve the migraine type of pain management. It is a wonderful drug. However, nowadays there are several type of semi-synthetic or synthetic derivatives are available with argolins, and it is also in current hot area about the psychoactive type of drugs which can release the cancer also. Okay, so cannabinoids plus argots, they can be utilized in combination to combat with dangerous cancerous third stage as well as second stage type of cancer. Likewise, cannabis, you can see from the slide how it's wonderful this plant is. It is also known as marijuana, and it is a psychoactive drug. However, it can definitely leads to addiction. So might be used cautiously as well as under the prescriptions of only our registered medical practitioners. And they can increase the appetite. Also, they can lead to the short term of memory. Sorry. Also, these alkaloids act as a pesticides and insect repellents. So one of the example is chrysanthemum. So chrysanthemum, in this, pyrethrum is a natural insecticide. It can be made from these dried flower, and they can be used as a insect repellent as well as pesticides in various types of formulations. Next category, third one is steroids and hormones. You can see from the slide that steroids are having the cyclopentanone phenanthrene ring system with their uh, elongated chain. So basically, it is an organic component which have four rings, that is A, B, C, D, and they have different type of biological components, active agents like cholesterol, estradiol, testosterone, dexamethasone, and likewise. Okay, so you can see that how cholesterol looks like. It is a structural component in our cell membrane and precursor for the biosynthesis of steroidal hormone, basically ergosterol. And ergosterol is found in the cell membranes of other microorganisms like fungi and protozoa. Similarly, progesterone, it is also called hormone of pregnancy, sex hormones involved in the menstrual cycles, embryogenesis, as well as lactation. Testosterone, it is helpful in the sperm production of secondary sexual characteristics, particularly in the male, promotes sexual behavior and motivation, and it is also detecting agent for doping among athletes to improve the performance. Okay. Next, other hormones examples are adrenaline and noradrenaline. They can be act as a stress hormone. Another name of these types of hormone is epinephrine, norepinephrine. So basically they have different type of physiological effects in the body, but at this moment I will say that when you see some aggression, when you see some flight or fight type of mode, this type of hormones can be released from our body, and they can be located at different parts, as majorly it is available in the kidney, and they can be released when you feel some sternus activity, or you have to perform some sternus activity, or you're facing some fight and flight or facing danger. So these types of hormones released from the body, they will increase heart rate, contract your blood vessels, dilates air passage, etc. Other therapeutic effect with these adrenaline and noradrenaline is they can be used in cardiac arrest, sometimes shocks and anaphylactic type of reactions, hypotension type of conditions, depressions, or in Alzheimer's disease. Next hormone is thyroxine that can be produced by thyroid gland, and it can be able to regulate your metabolism. And you will see how its structure. It is iodine containing component. So I'm not going with the general physiology. All of you just must be aware, because of biological sciences field, that how these types of nerve cells can release your different types of hormones or neurotransmitter that can be able to generate some effects. So you will see that how the synaptical vesicles looks like, where the neurotransmitter stores. When these types of stimulations exist, they can trigger this their release, and that can be uh, uh, interaction with the postsynaptic cleft, and they will affect the receptor actions. Now, the next molecules are, I categorize all these in the molecules of emotion. The words that has been generated by my very eminent Guruji, that these types of molecules they categorize in this category, that is molecules of emotions. That is dopamine, serotonin, adrenaline, mescaline, noradrenaline, P, as well as juvabayone. So how dopamine looks like, you can say it has been categorized with catecholamine because they have catechol ring in its structure, and they are responsible for your reward seeking behavior, that is highs of infatuation, learning, joy, self confidence, motivations. Okay, so all these types of physiological behavior are responsible due to the presence of this dopamine in our body. And if if it is a little amount, it will be a very joyful hormone that experience sense of adventures. Okay, that experience joy of life. Likewise, next is serotonin. that can be categorized in a mood molecule. It has been isolated in 1948, helps to maintain happy feeling. So very wonderful molecule. You can see from the structure, and you will see that it can be able to control sleep, make you calm, and relieving the depression. So naturally produced hormone, it is available and found in human brains, blood platelets, and digestive tract. Okay. So this is the general information. It is available in the literature. But my main focus is that you will know that all types of these hormones that are biologically exist have wonderful action in the uh, psychomotor activities as well as behavioral properties. So they are a good messenger, chemical messenger, or natural product chemical messenger, you can say. Another is LSD. Uh, we have already seen or heard in some movies, that is lysergic acid diethylamide. Short form is LSD, and this LSD is a very craving agent, and it can be used for addictive property basically. But in minute quantity as a drug, they have wonderful effectiveness in case of either pain, to calm your anxiety, in insomnic disorders, fatigueness, etc. Next is, or you can say, near about last molecule, that is oxytocin. And as you know that it is a mother hormone and little hormone. How it can be looks like, it is also a complex structure. You can say it is a nanopeptide hormone which is obtained from the pituitary gland and act as a neurotransmitter in the brain. So what are the various biological functions with oxytocin? They are urine contraction, uterine contraction, basically lactation, as well as pair bonding. Next or last category of this presentation is pigments. So the name is anthocyanin. It is a water-soluble pigment produced by the cytoplasm of the colored plant cells, and this is responsible to provide the pink red color of most of the petals of the flower. So how the molecules looks like, you can say it is a flavonoid type of derivative. Next, anthocyanins that can be responsible to appear red in color, purple, blue, deepening. That is depends upon the soil property, how the soil where these types of plants have been grown, and due to the presence of anthocyanin, they will give different type of color to the plant. They can also attract to promote the pollination process, also act as a sunscreen to absorb the UV light as well as antioxidant property. Next is carotenoids, another pigment. They are very long chain water soluble, not water soluble, water repelling pigment, which can be synthesized by plastids of plant cells. And you can see in the sunflower, the beta carotene is produced that can impart the bright yellow, orange color to the flower. So that is the pigment responsible for yellow color of the sunflower, and it is a biological natural products. Next is chlorophyll. All of us know that is responsible pigment for photosynthesis, and how this molecule looks like, it is represented in the slide. It is chlorophyll A. Next is palytoxin. It is a very complex marine natural products, and it has been isolated in 1971 by uh, uh, limu, that is which is present in some Hawaii, and they found that this component is the most toxic component known, and even 0.15 microgram per kg for responsible for death. So you can see from the structure how complex it is. It have 129 carbon atoms, 221 hydrogen atoms, 54 oxygens, 3 nitrogens, and 71 chiral center. And if anybody wish to learn about this molecule, if they have interest in the biological source, they can go for this reference that is available in Journal of American Chemical Society. However, this reference is very old, but it is available in the literature. You can see and you can read about this particular component. And in my personal view, you can take this natural marine product as a lead for further research. Brevetoxins, they are also in A and B. They are also a very complex structure with cyclic polyether component. They can be obtained with a species called Carenia brevis, and it has been synthesized synthetically in 1995, and that is the yield. Yield is very low. Next is Taxol. Taxol is a very wonderful drug for cancer, and it is extremely expensive because it cannot be prepared in a very easily way in semi-synthetic way. However, they can be harvested and obtained from the plant, that is Pacific U. So you can also read this type of total synthesis of Taxol. It is available in the journal, that is Nature, and you can date the break or more information from that. So that's all from my side. If you have any query, please let me know.
Thank you, sir. So the first question is, can we consider both primary and secondary metabolite as a natural product source?
Major one is secondary one. However, they are both categorized in the natural product sources, but secondary metabolites are easy comparatively to primary sources to synthesize semi-synthetically or synthetically.
Okay, sir. So the second question is, what is the best extraction method for terpenes?
Terpenes, nowadays, normal column chromatography can be used.
Okay.
However, whether hyphenated techniques are useful in various types of purposes basically. However, traditional methods are also very efficient if you wish to isolate them.
Okay,
But
sir.
hyphenated techniques are valuable given better results.
Okay, sir. Ms. Aman Patel, you can ask the question.
Okay. Sir, good morning. I'm Aman
Good
Patel from
morning.
the Department of Pharmacy, Sri Sai University. Sir, I have a
Yes,
very
ma'am.
basic question regarding the natural products. So, my question
Ji.
is, as if in today's life, natural products are in demand, everybody is going for natural products and herbal products. But, sir, there is a major
Yes.
issue regarding the standardization of that particular drug, herbal drug or herbal product we are using. So, sir,
Yes, yes.
is there any journal way for standardization as if we are using the herbal products in our day-to-day life, in our home as well?
Yes, definitely. You have raised a very good question, Aman,
Thank you.
and that is the genuine problem actually, because we have very few literature available right now about the standardization of the natural product basically. And however, Ayush department in India from Delhi, they are do lot of research and undergoing lot of research projects about these types of process in lieu with Indian Pharmacopoeia Commission, and they have maintained some good way of literature as well as standard data about them. So, however, it is not available for utilization in the home work, you can say, or home your native places. But you can do these types of things in our academic institution, uh, simple labs which have few facilities, not very you can say hi-fi strategies.
Okay, thank you, sir. Thank you so much.
Thank you.
So the next question is, do pherone act like a hormones?
That is that may be called as, but they do not have the structural moiety as a steroids. So hormonal activity in case of insects they can, but not in the case of human beings.
Okay, so the next question is, can you please tell us know about the plant alkali toxicity?
Basically 99% of alkaloids are toxic, but we have done a lot of research, our ancient scientists. So very minute quantity can be act as a drug. However, you can take these agents as a regular consumable item, they will definitely lead to the toxic.
Okay, thank you, sir. That was an amazing lecture. Thank you, sir, for sharing your great knowledge with us on chemical and biological importance of natural product. Thank you once again, sir.
Okay. So thank you, Dr. Amit, for inviting me in this wonderful session. And you have organized this 3 days conference in a very well mannered. I have, uh, you can say, listen all of their premier eminent scientists who are from the pharmacovigilance department, academic departments, as well as from pharmaceutical industries and other institutions. They have done a wonderful job. Best of luck for your team and future endeavors.
Thank
Thank
you
you.
so much, sir. Now, this is an announcement for all the participants that feedback form will be shared very soon. E-certificate will provide to those students who fill the feedback form. Now, we have a third speaker, Dr. Mohammad Azhar. He's working as assistant professor at K.R. Manglana University. He has over than 7 years of experience in research and development. He has presented number of papers at national and international level. He has also published books and delivered expert talk in different institutes. He is lifetime member of different professional bodies, and he's also a member of advisor of different journal. Now, I would like to call Dr. Mohammad Azhar to deliver his talk on the topic adulteration of mercury in skin whitening cream. Over to you, sir.
Thank you so much, Ms. Sharma, for giving the opportunity to speak on this platform. And right now, I'm sharing my presentation. Just let me know once it is visible to everyone.
Sir, now it's sharing. So screen is visible.
Okay, thank you.
Thank you.
Thank you, everyone, for giving the great opportunity to speak on this platform. And prior to this, I would like to give the highlights why I choose this topic. During the opening ceremony, I just listened the Honorable Chancellor Professor Rana regarding some of the skin whitening products, and he discussed about the mercury in skin whitening creams. So, thus, I plan to speak on this topic. This is a very hot topic abroad, but it is essential important for us as we are keen important into the skin whitening creams, and several times we are avoiding the detrimental effects of skin whitening creams. These are hard.
Excuse me, sir. Sir, can you please try for the slideshow?
Yes.
So now your screen is not visible.
Is this visible?
No, sir, now your screen is not visible.
Okay. Now?
No, sir. सर आप दोबारा screen share करो अपनी फिर maybe it will work. Yes, sir. Now it is visible.
Okay. And audio is, I'm audible?
Yes, sir. Yes, sir.
Okay. The topic is mercury in skin whitening creams, neurotoxicals as well as neurotoxicals. Today, I'm discussing about the harsh reality of Indian industry and why they are promoting this thing to other common people. Like in the next slide, you can see that these two pictures from the Bollywood and from the industry. Like the two actresses, the prominent actress in the Bollywood, like 20 years back, they have a different skin shades, and right now they have a very different shades. And we have a very renowned brand that is a Fair and Lovely. And after the criticism from the WHO and others celebrities for the colors, they remove the name from Fair and Lovely and thus become the Glow and Lovely. This one is set out to get rid of the words that propagated the ritual stereotypes. And the slide has introduction, the safety evaluations, methodology, and the results, rules and regulations are requirement for the mercury. and some publications, the media reports, and the references. As per the WHO guidelines and the WHO data statistic states that the skin lightening industry is one of the fast growing beauty industries in the world and approximately 31.2 billion US dollar in the US. And what is the concept behind it? The whitening creams are being used to achieve the fair or the spotless skin. Fair colors were considered the aesthetic symbol of personality, and this misconceptions energize the harsh hues of whitening creams in across the globe. Why the mercury is used, the basic mechanism of actions, how the mercury acts. Mercury salts block the synthesis of melanin as it competes with the copper in the actions of enzyme tyrosinase. That is the main mechanism of actions. And the melanin is synthesized in the melanosomes and deposits inside the keratinocytes, thus cause the blackening of the skin. And second reason is the economic importance. The mercury is being very, very economic, very, very cheap. Thus, the industrial use of mercury in skin whitening cream. So, do we check? Do your skincare products contain mercury? Do we ever check in our life in our products on daily basis, like cold creams, skin whitening creams, or any skincare products topically? And do we discuss about the use of mercury in any products? Hardly we do this. The bitter realities but exist in our country where the arranged marriages are still the norm. The matrimonials ads consistently described of women's complexions and a dark-skinned women often pay a higher dowry. That is a very bitter realities in Indian scenario. The bullying and taunting of dark-skinned girls and women is common. Why? Dark-skinned actors complain of fewer roles. The next is the advertising campaigns of various brands have typically depicted women and increasingly men as winning better jobs and partners thanks to the fairness claims. We can evidently some of the major advertisement promoted by the Hindustan Unilever Limited. And the sumoto actions has been taken by the ACC, and the Advertising Standard Council of India's on the Consumer Complaint Council upheld complaints against the 200 advertisement. A total of 3,119 cases were brought up to ASCI notice, and sumoto actions was taken against the 148 advertisement. This is a very latest example of Hindustan Unilever Limited promoting the Ayush, Haldi soap, saffron soap, and cow's heaps. This was I'm highlighting some of the facts over here that were inadequately substanced and are misleading. The other product is the Sarvadya Skin Hair Clinic. They are also promoting the successful treatment of the disease like a big white spots was considered to be primary facie in violation of DNC rules and the DMR Act. A total of 319 cases were brought to the ASCI's notice and sumoto actions was taken against 148 advertisements, and the rest being complaints through the direct sources. Out of the 200 advertisements against which complaints were upheld, 82 belong to the healthcare, 75 to education, 11 to personal care, and 8 to the food and beverages categories, and 25 to other categories. Our obsession with the fair skin did not come from the HU or the Imami. It is deep-seated cultural bias and equates the being fair with the being superior. Told by the Thomson Reuters Foundations. When a demand for a product exists, a manufacturer will cater to that demand, and the people will buy that product even without a Shah Rukh Khan or John Abraham telling them to buy. This is some of the prominent role of the melanin. What is the role of melanin in skin? It's protection against the UV, in eye protection against the UV and high frequency of visible hair, antioxidant defense for the skin, capability to trap or bind the chemicals, neural cells protection against the toxic compounds, inner ear. And now using the whitening creams or any cosmetic products, we are removing our melanin. That is a defense mechanism to provide the natural system to our body. We are removing these essential component from our body and making our body to be fair. These are some of the adverse side effects of mercury in GIT and the kidneys. The protein digestions inhibitions is caused by the mercury. Mercury has selective destructive effects on the intestine, especially large intestine, where it passes to be excreted, leading to intestinal ulcerations. Resulted in abnormality with GIT. In kidney, cumulative accumulations cause the acute tubular necrosis, death of tubular epithelial cells that form the renal tubules of the kidneys, and apart from it, cause the nephrotic syndrome, edema, and the loss of the protein from the plasma into the urine due to the increased glomerular permeability. This is the effect of the mercury on pregnancy, how it affects the fetus. Mercury causes the inhibition of the cellular enzymatic process by binding with the SH in amino acids, appears to be a major part of the connection to allergic immune reactions. On expressed to cellular level, it altered the cell permeability, changes in the macromolecular structure due to its affinity for sulfahydryl and thiol group, and DNA damage. Affect the children who are on the breastfeeds and whose mothers are using the mercurial cosmetics, thus the toxic to children as well. This is like outsource it the pamphlet shared by the FDA about the skin products across the world. This is a methodology that we use during the estimation of mercury in our lab. The instrument is use the ASK, the atomic absorption model built by the Agilent Technologies, and the methodology adopted earlier used by the CTAC technology of USA for the estimation of mercury in skin whitening cream. This is the results that we conduct in 11 commercial available products. Like mostly people use the Fair and Handsome, Pond's Bright Beauty, Garnier Skin, Fair and Lovely, Olay, Vaseline, Fair and Lovely SPF 15, Lakme, Jobies, Lotus Herbals, and they contain these kind of mercury. This is the first year result, and I'm going to present the second year results. So here we have the results. In the second year, we can see the Fair and Lovely 50 grams contain the mercury 70.75 ppm parts per million. And next is the Lakme 925, and apart from it, Jovis. The violation of law and drugs cosmetic rules 1945, the drugs and the magic remedies objectionable advertisement act 1954, drugs and cosmetic act 1940, and rules 1945. According to these sections 145D, prohibition of manufacture of cosmetic containing the mercury. No cosmetic containing mercury compound shall be manufactured. And as per the Bureau of Indian Standards, as per the Part 2, list of raw materials, particularly the raw materials generally not recognized as safe for use in cosmetics. Mercury and its compound, except those special cases included in Annex C. So it's our responsibility to check about the labeling of a product. Nivea recently put an ad in the Middle East with the tagline "White is purity." However, it is changed uh, after it was slammed as a racist. Similar products are on sale in Southeast Asia, the Middle East, and in the parts of Africa. Although India, with its population of 1.3 billion population, it is its prized consumer market. And apart from it, Ghana, that is a very albinism country, Ivory Coast, Ghana, have banned the cosmetic containing hydroquinones, which critics say can cause a cancer. A similar ban exists in the Japan, South Africa, and the European Union. This is the publications published in last few years, and more publications are into the pipeline. The same present another kind of presentation has been presented in the Ministry of Defense, DRDO in Delhi. And next is I'm going to present some of the impact of our research has been presented by the Z News, major news channels in India, and apart from it, by publishing by several time in several newspapers in print media. These are some of the conclusions. It has been reported by the WHO that the skin whitening creams contain the mercury to toxicity levels Other reports published by the FDA concluded that the 11 out of the 15 samples contained the mercury levels more than 1 ppm and banned by the WHO. There is no proper labeling about the mercury contained in most of the cosmetic products. Attention must be paid to monitor the presence of mercury in cosmeceuticals and for its truthfulness in their labeling on the wrappers. Regulatory agencies must frame the rules and the regulations to have a control on the mercury content. There is no uniformity for mercury limits for such products by the WHO, FDA, and the DCGI, particularly for India. These guidelines need to be uniform, and all regulatory agencies should implement the mercury limits. Besides that, there is a lack of awareness among all the active as well as the passive consumers of the cosmetics, such as skin whitening creams. Apart from it, government and the private health agencies should initiate some public awareness program. Basic research startup, honey adulterations, desi antibiotics, all are adulterated with some amounts of antibiotics or some previous amount. As this year we are hosting the presidency for G20, it's our responsibility of the scientific fraternity to raise the standards of health and healthcare products among the India, and we should present our global pictures. So, yeah, these are the key points, key message, awareness among the common people with scientific research, promote the young researcher towards the vigilance health program, labeling of the cosmetics, herbal formulation, nutraceuticals, the false claim are very prominent these days over the name of the Ayurveda and others skin care products. Reporting and what we can do in this, the reporting the misleading advertisement to the Advertising Standards Council of India and report to the BS, Bureau of Indian Standards. Thank you.
Thank you so much, sir. That lecture is just incredible. I appreciate your sharing your vast knowledge with us, sir. We thank you so much, sir, once again.
Thank you.
Thank you, sir. We have shared the feedback form. Please kindly fill the feedback form. E-certificate will provided only those students who fill the feedback form only. And certificate will be generated by next week. Now, I would like to give a brief report about our old sessions. One day, on a first day, Professor Dr. Santosh Kumar Verma was the first speaker. He delivered his talk on the topic biomarker of toxicology. Sir has explained biomarker in toxicology furnished indication on the effect of toxins upon a specific target, where cell organ or a system, and they can be biomarker for exposure susceptibility or effects. Sir has also explained biomarkers and mechanism of toxicity. Second speaker of the day one was Dr. Shamim Ahmed Bande, who shared his knowledge on the topic, why do we conduct preclinical and clinical studies? Sir has explained the importance of preclinical and clinical studies for developing novel diagnostic and therapeutics, and the result of experiment on animal models of disease often inform the decision to conduct studies in human. He also explained preclinical studies are used to define pharmacological and toxicological effect, while clinical trials are used to generate safety and efficient data for human health. On day second, Dr. Harminder Singh was the first speaker. He delivered his talk on the topic cell phone radiation and human health. Mobile phones are at the high risk of exposure to electromagnetic field due to the MIMO system, which can interfere with the body's on electromagnetic energy system, causing stress and adverse health effect. Sir has also explained children are more prone to due to their smaller head and brain cells, thinner cranial bones and skin, thinner more elastic ears, lower the blood cells volume and greater conductivity of nerve cells. Second speaker of the day, second speaker of a day two was Dr. Vijit Agarwal, who shared his knowledge on the career opportunity in pharmacovigilance. Sir has mentioned that pharmacovigilance has a significant shortcoming, including the accuracy of reporting adverse drug reaction, a lack of reporting, and others. However, pharmacovigilance efforts continue to be a crucial discipline in the management of unfavorable consequences of developing medical goods. A variety of increasing effectiveness health products with an essential and often unpredictable tendency to cause side effect, raising safety and concern. He told us how they affect the key pharmacovigilance component that must be taken into account during the analysis and management of adverse drug reaction. Third speaker of the day, Dr. Hitesh Kumar, who shared his knowledge on the molecular basis of steroids biosynthesis in stevia, a source of non-californic sweetener. Stevia is an economically important plant species due to the presence of high concentration of sweet taste compound, steroid glycoside in its leaf. Steroid glycoside are approximately 300 times sweetener than common tablet sugar, used as non-californic sweetener in many countries of the world. Sir has also told us the cloning function and characterization and expression regulation of a various gene involved in steroid glycoside biosynthesis. In addition, he also told us cloning and analysis of promoter of isolated gene will be addressed. Now, may I request Dr. Meenakshi Sharma, Organizing Secretary, to propose a vote of thanks. Over to you, ma'am.
Thank you, Ms. Pritancha. His Excellency, Honorable Chancellor, Engineer S.K. Punj, Chief Managing Director, Ms. Tripta Punj, Pro-Chancellor, Engineer Tushar Punj, Honorable Vice Chancellor, Professor Dr. R.S. Rana, Pro-Vice Chancellor, Professor Dr. N.N. Sharma, Registrar, Professor Dr. Zahid Ali, speakers of the event, Professor Dr. Santosh Kumar Verma, Dr. Shamim Ahmed Bande, Dr. Vijit Agarwal, Professor Dr. Mahinder Singh Ashawat, Dr. Hitesh Kumar, Dr. Harvinder Singh, Professor Dr. Girish Kumar Gupta, and Dr. Mohammad Mazhar, Deans of the University, faculty members, dear students and participants. A very good afternoon to one and all. Someone well said that gratitude is a powerful catalyst for happiness. It's the spark that lights a fire of joy in your soul. Feeling gratitude and not expressing it is like wrapping the gift and not presenting it. It gives me immense pleasure to stand in front of you to propose a vote of thanks on behalf of Sri Sai University, Palampur, as organizing secretary of the Symposium, Trends and Innovation in Pharmaceutical, Biochemical and Biosciences, organized by Sri Sai School of Pharmaceutical Education and Research. Sri Sai University, Palampur, sponsored by PharmaLaw. The main focus of the National Symposium is to provide a global furnished platform for all researchers and academicians related to pharma and biosciences to share their opinions and learn about important worldwide breakthrough developments in health sciences. On behalf of Sri Sai University, Palampur, first of all, I take the opportunity to express my gratitude to His Excellency, to our Honorable Chancellor, Engineer S.K. Punj, for his support for every new step forward. Under your forward-looking leadership, Sri Sai University feel encouraged and motivated, sir. I extend my special thanks to our Chief Managing Director, Ms. Tripta Punj, for her support and cooperation. Thanks, ma'am, your guidance in our difficult time, and always encourage us to cross the finish line. I would like to convey my deep gratitude and hearty thanks to our Pro-Chancellor, Engineer Tushar Punj, for his all-time suggestions and support. We thank you, sir, for trusting us. I take this opportunity to present my deep thanks to our Honorable Vice Chancellor, Professor Dr. R.S. Rana, for gracing the inaugural ceremony and sharing his words of wisdom. I'm immensely thankful to our Pro-Chancellor, Professor Dr. Anand Sharma, for his enormous support and constant source of motivation. A special thanks to our Registrar, Professor Dr. Zaid Ali, for his enormous cooperation within the organization for this gala event. I extend a really hearty vote of thanks to our speakers, Professor Santosh Kumar Verma, Professor Dr. Mahendra Singh Ashawat, Professor Dr. Girish Kumar Gupta, Dr. Vijay Agarwal, Dr. Shamim Ahmed Bande, Dr. Harvinder Singh, and Dr. Hitesh Kumar and Dr. Mohammad Mazhar, who spared time from their busiest schedule to grace the event today. We had opportunity to hear your thoughts, and this will surely be going to encourage us in our future events. Thanks, sir, for sparing your precious time and sharing knowledge to the gathering. With deep sense of appreciation, I would like to extend my thanks to our convener, Dr. Amit Sharma, who always motivated us and stood as pillar of strength. Thanks, sir, for your availability 24/7. A special thanks to our technical advisor, Professor Dr. R.P.P. Singh, Team Engineering, and his technical team members, Engineer Sandeep Rawat, Engineer Shivanshu, and Engineer Nandini for their selfless efforts. We will remain grateful to you for your technical support and help. Heartfelt thanks goes out to our organizing secretary, Ms. Pratancha Sharma, for her valuable contribution and enormous efforts. I'm short of words for her involvement and her willingness to take in completion of tasks beyond her comfort zones. I extend my thanks to Symposium coordinator, Ms. Aman Patyal, and organizing team members for their efforts to make this symposium a success. Our special thanks to Pharma coordinator, Mr. Amit Shah, and technical team members for sponsorship and coverage. My deep sense of thanks and appreciation goes to all the participants for attending the symposium with great enthusiasm and made it a great success. Thank you all so very much for being with us for the entire session. Have a wonderful day ahead.
May I now request to everyone, please switch on your cameras for a screenshot. Please, all the member of this Google Meet, please switch on your cameras for a screenshot.